Immunotherapy changed cancer for some people, but for most colon cancer patients, it never worked. Until now. A new approach finally showed results for the most common type of colon cancer, and the follow-up data made it even more surprising. In 2024, researchers at one of the world's leading cancer centers, Dana-Farber Cancer Institute in Boston, published results in one of medicine's most respected journals, Nature Medicine, announcing something nobody expected: the first safe and effective use of immunotherapy for the most common subtype of colon cancer. This is that story. To understand why this breakthrough matters, you need to understand the enemy. Colon cancer, or colorectal cancer, is the fourth most common cancer in the United States and the second leading cause of cancer death. Every year, roughly 150,000 Americans are diagnosed. But not all colon cancers are the same. Buried inside the genetic code of every tumor is a clue about how it behaves, and crucially, how it might be beaten. About 3 to 5% of colon cancer tumors are classified as MSI high, or microsatellite instability high. These are tumors with a kind of genetic messiness. They have thousands of tiny mutations scattered throughout their DNA, and crucially, those mutations act like flags, bright red flags that the immune system can see, recognize, and attack. That's why, when immunotherapy came along, drugs that essentially unlock the immune system and point it at the cancer, MSI-high tumors were vulnerable. The immune system already knew what it was looking for. Then there's everyone else, the other 95 to 97%. These patients have what's called microsatellite stable, or MSS, colorectal cancer. Their tumors are genetically tidy. They don't have all those mutations, and they don't wave those red flags. To the immune system, they look almost normal, which is exactly the problem. MSS colorectal cancer is what doctors call a cold tumor. It's invisible to the immune system. And for years, for decades, that meant immunotherapy was essentially useless against it. The principle behind immunotherapy, specifically immune checkpoint inhibitors, is elegant. Your immune system has T cells, tiny warriors trained to hunt and destroy threats. But cancer is clever. It uses proteins called checkpoints. Think of them as off switches to tell T cells, stand down. This isn't a threat. Checkpoint inhibitor drugs block those off switches. They tell T cells, ignore the off signal, stay in attack mode. And in MSI-high cancers, this works beautifully because the T cells can find the target once unshackled. But in MSS cancers, there's a deeper problem. Even if you remove the off switch, the T cells still can't find the tumor because it's disguised and it barely looks like cancer at all. The immune system is willing. It just can't see the target. That's where Dana-Farber's work comes in. Dr. Benjamin Schlechter, a gastrointestinal oncologist at Dana-Farber Cancer Institute, co-led a clinical trial testing a new combination of two next-generation immune checkpoint inhibitors, Botensilimab and Balstilimab, developed by the biotech company Agenus. Here's why this wasn't just another rehash of old immunotherapy drugs. Botensilimab, or BOT for short, is different from previous checkpoint inhibitors in a few critical ways. First, it targets the CTLA-4 protein on T cells, just like older drugs, but targets a different region. It's been engineered to physically bind tighter inside the tumor, making T cells extraordinarily active exactly where they need to be. Second, and this is what makes it special, it recruits a completely different class of immune cells, natural killer cells, or NK cells. These aren't T cells. They're a separate line of immune defense, and by bringing NK cells to the tumor environment, Botensilimab does something previous drugs couldn't. It changes the environment around the tumor from immunosuppressive to attack-ready. Balstilimab, the second drug in the combination, targets a different checkpoint protein called PD-1. Think of it as the second lock on a door. BOT picks the first lock, BAL picks the second. Together, they throw the door wide open. As Dr. Schlechter described it, a small tweak in a known pathway can make a big difference in patients. That turned out to be an understatement. The trial was a phase 1 study, meaning its primary goal was to assess safety, not necessarily to prove the drug works. But what they found went far beyond what anyone expected. 142 patients with MSS metastatic colorectal cancer were treated. These were heavily pre-treated patients. Many had already been through 3 or more lines of chemotherapy, including drugs like regorafenib. Let's put those numbers in context. For metastatic MSS colorectal cancer patients who have failed multiple lines of therapy, median overall survival historically sits somewhere around 5 to 7 months. This combination produced a median overall survival of nearly 21 months and a 42% 2-year survival rate. That's a survival plateau. People who responded didn't just survive a little longer. Some of them appear to be in durable remission. 73% of patients who benefited had some form of clinical activity from the treatment. Nearly a quarter had a major response. And critically, the median duration of response was over 16 months. These aren't fleeting responses, they're lasting. In the words of Dr. Schlechter himself, these results show the kind of meaningful, long-lasting benefit we rarely see in this setting. Now, before hope turns into false expectation, we need to talk about who this currently helps. Because there's an important caveat in these results, and it matters enormously. This combination, in this trial, worked specifically in patients without active liver metastases. That detail changes the math significantly. Colon cancer is notorious for spreading to the liver. A large proportion of metastatic colorectal cancer patients, many estimates put it above 50%, have liver involvement. And we know that the liver has a unique immunosuppressive environment. It actively dampens immune responses, which is why in this trial, patients with active liver metastases showed far less benefit. So, who does this currently apply to? Metastatic MSS colorectal cancer patients who have liver metastases that have been surgically removed or ablated with no active disease, or who have no liver metastases at all. When you apply those filters, you're looking at a meaningful but specific subset of patients. So, you're potentially looking at tens of thousands of patients in the US alone who could be eligible for this treatment right now through clinical trials, and potentially far more once it reaches FDA approval. Researchers also note that patients with RAS mutations, which appear in around 67% of MSS-CRC patients, still responded. This isn't a treatment that requires a perfect genetic profile. It's broadly applicable within its eligible population. And while that's the headline from the metastatic side, there's a second major breakthrough worth knowing about. This one for a completely different subtype and a completely different stage. Presented at ASCO 2025, the world's largest oncology conference, Dana-Farber's Dr. Jeffrey Meyerhart announced results from the ATOMIC trial. This was a phase 3 randomized study for patients with stage 3 colon cancer, disease that spread to nearby lymph nodes but not to distant organs, with a specific molecular characteristic called deficient DNA mismatch repair, or DMMR, also known as MSI high. Remember that 3 to 5% of colon cancers, this is them. And in the ATOMIC trial, adding an immune checkpoint inhibitor called atezolizumab to standard adjuvant chemotherapy after surgery reduced the risk of cancer recurrence by 50%. That's potentially curative for a population that historically has had significant recurrence rates. So, to summarize, we now have powerful new immunotherapy data for both ends of the colon cancer spectrum. For stage 3 DMMR patients, a 50% reduction in recurrence risk. For metastatic MSS patients, the group that previously had nothing, a 42% 2-year survival rate and ongoing durable responses. Science is always honest about its limitations. First, liver metastases. This is the biggest unsolved problem. Researchers are actively working on combinations that might extend the benefit to patients with active liver involvement. A phase 1 trial testing BOT and BAL combined with FOLFOX chemotherapy and bevacizumab showed an astonishing 71% objective response rate in 14 patients with MSS metastatic CRC. Early data only, but it signals that combining immunotherapy with chemotherapy could unlock the liver problem. Second, we don't yet have good biomarkers to predict who responds. About 20% of patients had a confirmed response, but we don't yet have a reliable test to know in advance which 20% you'll be in. That's a critical area of ongoing research. Spatial biology analyses are being conducted to understand the tumor microenvironment in responders versus non-responders. Third, phase 3. Everything we've discussed from the BOT-BAL combination comes from phase 1 and expanded phase 1 data. The BATMAN trial, the global phase 3 registration trial, started in Q4 of 2025. That trial will determine whether these drugs get FDA approval. The FDA has already aligned on the trial design and waived the need for a monotherapy control arm, an unusual step that signals genuine excitement at the regulatory level. Fourth, access. Right now, this drug combo is only available in clinical trials. The phase 3 BATMAN trial will be a global 2-arm study. Patients with refractory MSS, metastatic CRC without active liver metastases will be randomized to receive either the BOT-BAL combination or the standard of care. This is the study that will take this drug from promising to approved. And if the phase 1 data holds, if the 42% 2-year survival plateau proves durable and reproducible, this combination could fundamentally transform how doctors treat late-line colorectal cancer. Agenus is also pursuing expedited regulatory pathways, including accelerated approval and real-time oncology review, mechanisms that could shorten the timeline between trial completion and patient access. The story of colorectal cancer and immunotherapy is, for the first time, no longer a story of failure. It's a story of a locked door that brilliant researchers refuse to stop trying to open. If this video helped you understand what's happening in colorectal cancer research, share it with someone who needs hope today. Subscribe for more science-driven health videos and drop a comment below. Did this change how you think about immunotherapy?